This video is going to overview the function of the eye. Certainly around the eye, there's a number of structures for protection. There are eyebrows and eyelashes. This prevents the entry of foreign objects. There are eyelids, which can be blinked and thus help protect the eye here. And then there are lacrimal glands on the lateral part of the eye, which secrete the tear solution, which then flows medially into that small opening, the lacrimal duct of the lacrimal bone. So a number of things on the exterior. There are also muscles. Now, in the embryonic head, it's thought that there were like different segments of muscles that then break down into three segments which contribute to the eye muscles, which are the most conserved muscles in vertebrates. The first of these segments produces four muscles. And as we'll see, one of the cranial nerves, the ocular motor nerve, innervates these four. Then the middle one produces the superior oblique muscle, and it's another cranial nerve, the trochlear, which innervates this one. And then it is the abducens nerve, which then innervates the final single muscle, the lateral rectus, which comes from the third segment. And so these muscles then move the eye. The superior rectus can move the eye superiorly. So when we pull on the eye from above, it moves up. There's an inferior rectus for down. There's a medial rectus for focusing on closer to the midline, a lateral rectus, and then the obliques, the superior and inferior, can cause the eye to rotate. So we have these muscles. And as you may remember from the study of the cranial nerves, there are then three cranial nerves whose primary function is to move the eye. Two come from the midbrain, the ocular motor and the trochlear, while the abducens comes from the pons. So cranial nerves 3 and 4, the ocular motor and the trochlear, are coming here from the midbrain. Cranial nerve 6 from the pons. All of them pass through the superior orbital fissure to reach the region of the eye. And as you may remember, like if you were ever to, fall and hit your head or something, someone might say, here, can you follow, my finger with your eye? When you do that, you're testing three different parts of the brain. So if there was inflammation, if there was damage, then you would notice a change in the ability to move the eye. So you can actually just check three different cranial nerves, three different regions of the brainstem by doing that. These nerves, in addition to moving the eye, can also do a couple of other things, like allow us to focus, control the eyelids, so drooping of the eyelids might result if these areas are damaged. And I mentioned that a bit in this video, just going through, some of the things that that damage might result in. When we consider the eye, it's typical to look at it as a series of four layers, and that includes the lens. So the outermost layer is what we call the fibrous tunic. So this includes the white of the eye, which has collagen. Now, collagen is typically light in color. Obviously, we can see it here. But if you were to develop scar tissue, that's lighter in color. So collagen is lighter in color, making up the white of the eye here. And this is what makes it very tough. If you've ever dissected in biology class, like a sheep eye or a cow eye, you may remember that you actually had to push really hard through the to get through the fibrous tunic of the eye because of all of the collagen there. And so we have collagen making up most of this fibrous tunic. In this region, the white of the eye, we call the sclera. But obviously, if that's all the eyeball was, we wouldn't be able to see, all right? Because obviously there would be no place for light to come through. And so here we have a transparent region in the front of the fibrous tunic known as the cornea. Now, I know this sounds odd, but some of your cells are transparent, like glass. Just because you can see through it doesn't mean it isn't there. And the same thing is true of your body cells. The cells of the cornea and the cells of the lens are so constructed so they do not interfere with the passage of light. And so they have proteins like crystallins. But once again, these proteins don't interfere with the passage of light. So part of you is see-through the cornea and the lens. Once again, if you ever dissect, say, a sheep eye or a cow eye, you'll notice that after death in the preserving material that the proteins will change their shape. And so now you'll see the cornea, it will look more of a bluish color. So the outermost layer of the eye is this fibrous tunic here, which includes the sclera and the cornea. The second layer is what's called the vascular tunic, because this is where the blood vessels are. And I just, it's kind of interest in that usually if you're seeing someone's blood vessels, they've hurt themselves. All right, so under normal conditions, we don't see blood vessels. But actually, the eye is the exception because the cornea and lens are transparent. If you look through them, then you can actually see the blood vessels of these of the vascular tunic. And, for example, if you were suspecting, diabetes, or, there's reasons that looking at someone's blood vessels, might, give you something about information about their health. So looking through the eye and looking at these blood vessels can be of value. Most of this vascular tunic is known as the choroid. It is black in color. So we make melanocytes, not just for hair and for skin, but also in other parts of the body, like melanin, is produced in the brain and the substantia nigra as, an intermediate in neurotransmitter synthesis. But here you can see, a very black pigment here. Why? Well, let's say that, I would like to detect light. Well, the light, I don't want it, if it isn't absorbed by a rod or a cone, I don't want it bouncing around the eye. That would confound vision, because if light is bouncing, I don't know whether the light I just saw was something from an object in front of me or something that's bouncing around. And so therefore the melanocytes here and their melanin absorb this light that isn't absorbed by the retina. So this is deep to the retina. So the retina has first chance of this light, but if the light doesn't get it, if the retina doesn't get it, then it gets absorbed here. Okay. And so the choroid makes up the majority of this vascular tunic. But in the anterior part of the eye, we have some specialization. So there's what's known as a ciliary body, which has the ligaments which bind the lens. And so the lens is going, as we'll see, to change its shape. There's sometimes it gets stretched, and then sometimes it's relaxed and gets fatter. So the lens can be pulled thinner to focus on something that's far away, or be allowed to relax and get fatter to look on something that's close. And what does this is the tension in the ligaments in the ciliary body. And there's a ciliary muscle in this region, which then pulls on these ligaments. So you can pull on these ligaments and thus change the shape of the lens. Anterior to the ciliary body, here I've colored it brown, is an area known as the iris. This is the pigmented part of that eye that could be brown or green or blue. And it is the smooth muscle here which controls the diameter of the pupil, the opening which allows light to enter the eye. It's smooth muscle, and you know that because if you try to make it move on purpose, so if you say pupil constrict, you can't do that voluntarily. So it's not skeletal muscle, which is under voluntary control. This is smooth muscle, so this is an involuntary reflex. And this is good because, when you're, walking at dusk or at night, if there's starlight or moonlight, you want to let the maximum amount of light enter your eye. Otherwise, you wouldn't be able to see and you would trip and fall. However, if you were out on a bright sunny day, like imagine, especially like in winter when there's snow, it's a bright sunny day, but there's all this light that's being reflected off the snow. If your pupil was wide open, that would be too much light. And once again, you wouldn't be able to see, or you would fall because that's too much light. And so then you would want your pupil to constrict. And so to see optimally, we don't want too little light nor too much light. And so we can change the diameter of the pupil to allow for the greatest amount of light. Then next the lens, and I apologize when I first started. So there are 3 layers of the eye. I said 4, but I was in, so I just know there's 4 things I want to talk about. So 3 layers and then the lens. I apologize. So the lens is here being held by the ligaments. And so there are 2 transparent parts of the eye. There's the cornea here, that's part of the fibrous tunic. And then here you have the lens being held behind the pupil by these ligaments in the ciliary body, and the ciliary muscle can change the tension on these ligaments and thus change the shape of the lens. Now, before I talk about the retina, which will be the remainder of the discussion here, this space here is called the anterior chamber, say between the iris and the cornea, the anterior chamber, and it's filled with a watery fluid, the aqueous humor. Okay, there's also a small posterior chamber behind the iris and between the lens, but I don't really focus on that. So the anterior chamber here, it has this aqueous humor. And this aqueous humor is important because it has to nourish the cornea and the lens. So once again, I said that they're transparent, just so one, comment on that. They don't have blood vessels. They don't have capillaries. And I ask my class, I say, look at me. All right, now, if you're going to summarize what you see in one word, and I don't want to know what that word is, think of that word, and I say, okay, fine. Was anyone thinking of the word pink? And I say, all right, well, if you weren't thinking of the word pink and I've proven something, you don't have blood vessels in your cornea or lens. If you did, if there were blood vessels there, I would be pink or red. And so would the sky, and so would the grass, and so would the wall. Everything would look pink to red because you would be looking through a layer of blood. All right, obviously we don't want to do that. So we don't have blood vessels here. But as a result, these are cells, they're alive. How do they get their oxygen, et cetera? Well, this aqueous humor then is very important in providing the nutrients and the gas exchange for these living cells. They are alive. They get this then from diffusion. So the cornea and the lens would be examples of tissues in our body which don't have a blood supply. Okay. So that's the aqueous humor in that anterior chamber. Most of the eyeball is composed, it forms this chamber known as the vitreous chamber that's filled with a jelly-like material called the vitreous humor. All right, so it's jelly-like, and this helps to maintain the shape of the eyeball. Now, this fluid, we don't want the pressure to be too great because the retina, which I'll introduce in a second, it isn't really well attached to the vascular tunic, and too much pressure in the eye can actually cause the retina to separate. And so the innermost layer is what's called the neural tunic, the retina, and it is here that the cells which are detecting light are found around rods and the cones. So when we consider the anatomy of the eye, there are these 3 layers and then the lens. And once again, I apologize at the beginning, I said 4. So there's the fibrous tunic that makes up the sclera, which has all of the white collagen fibers, and the transparent cornea, right? So that's the outermost fibrous tunic. There is a middle vascular tunic that has the choroid, where blood vessels are, the melanocytes here absorb light. There's the ciliary body, whose ligaments control the diameter of the lens, and then the iris, which controls the diameter of the pupil. And then there's the innermost layer, the retina, where the nerve cells are. So to perceive light, we need to bend it. All right, because like if you're going to look at a building, obviously you don't have a size of your retina, comparable to the size of a building. If the light were just, coming straight, that's what you would need. So you have to bend light so that light then becomes in focus and forms a little picture on your retina. There are two parts of your eye which bend light. Your cornea is always bending light. So this curved cornea is bending light, and it does actually most of the bending, maybe 3/4 of the bending. The second structure is the lens. The lens is adjustable because of those ligaments I had just mentioned. And so when you are going to then bend the light so that it can focus on the retina, then once again, the bending occurs in two places. One, the cornea, which does 3/4 of the bending but is not adjustable, and then about 1/4 of the bending occurs at the lens, which is then adjustable. Now, the reason we want to adjust it is because let's say you're looking at something that's far away, like that cow. Well, the light that hits the cow and then bounces off the cow to hit your eye, it's already the light rays are kind of coming in parallel. If they weren't, then they kind of bounced in a different direction. All right, so light bounces off the cow in all directions, but because it's far away, the only ones which reach you are the ones which are coming in parallel. So the cornea bends this light and the lens bends it, but it turns out you don't need to bend it all that much. And so what we want when we're looking at something that's far away is then to make the lens narrower. We don't have to bend it too much. So we want the light to pass through less lens. So you then pull on those ligaments, which pulls the lens narrower. And thus the light from the cow comes into perfect focus on the retina there. Now, if you were looking at something that was close, well, then the opposite would then be true, because this light is still spreading out. So if you put something in front of your face, the light that's bouncing off here is not all coming parallel. It's still spreading out, all right? If you want to focus it on the back of your retina, you have to bend it when it's going parallel. But if it's doing this, all right, to make it then do this, the opposite, you have to bend it even more so that here the cornea is going to bend the light again. But then when it comes to what the lens does, the lens is going to have to bend the light more than it does when we're looking at something which is far away. So now we would want to relax those ligaments. The lens becomes fatter, and then as the light passes through more lens, it bends more. Now, just a quick comment on glasses, but obviously that's a whole nother scenario. So why might you need glasses? Well, if you have something known as myopia, this could be caused by having a thicker lens or an elongated eyeball. Okay, so if your lens is thicker, or if your eyeball is too wide, you bend light too much, okay? And so therefore, you are what's called nearsighted. It's easy for you to look at things which are close because you're good at bending light, all right? But it's harder for you to see things which are far away because you bend light too much. Right? You don't have to bend the light from far away objects all that much, but you are. And so you are nearsighted. You're very good at seeing things which are near. But you could correct this with glasses that would then take this light and then spread it out just a little bit. So let's say that light from the cow is coming into my eye. If I spread it out just a little bit, if my eye is bending light too much, if I take the light coming in parallel, I spread it out a little bit. Now it turns out I bend it just enough. If, however, I am farsighted, which is very common as we age, as your pupil becomes less elastic to become fat. So I am far-sighted. I need glasses to see things which are close. The reason, so you might have this from birth. Once again, if your eyeball is if your lens is too narrow, if your eyeball is too short, or once again, if it, the elasticity is less and it doesn't get fat. So if you can't bend light enough because your lens isn't fat enough, your eyeball is too short, then when you look at something which is close, you can't bend it. You want it to become perfectly in focus here on the retina, but it would actually be more in focused here. So now you need glasses which actually do a little of the bending for you. So they do a little of the bending of light. So your eye then has to do less. Okay. So the cornea and the lens are then important to bend light. Now let's talk about the perception of light, and this occurs in the retina. Here we have two cells which have modified the primary cilium. So all cells have a primary cilium. But interestingly, this cilium can actually be sensory. So in olfaction, for example, the primary cilium is sensory. Here in the eye, it is this primary cilium which then has the molecules which will interact with light and allow us to detect light. In some of these cells, this area is rod-shaped. These are called rods. In other, the outer segment. In other areas, it is cone-shaped, and we call that a cone. Now, because we're primates and higher primates that depend on life, in the day, we talk a lot about color vision and vision which occurs in bright light. But the majority of mammals are actually nocturnal. And so rods have the ability to detect light in fainter light, but they do not have the ability to see in color. So at night, you see, light, dark, and shades of gray. You're not seeing colors because at night you're using your rods rather than cones, which need bright light. And so we're using rods. Now, when I look straight at something, the light that I look straight at hits an area of the retina known as the fovea. So the light that from the object I'm looking straight at hits the fovea. And this is where the cones are located. The rods are located lateral to that, around the fovea. So you may notice at night, if you want to say, look at a distant star, so like it's a faint star, don't look at it, look next to it. If you look at a distant star, the light from the star hits the fovea, all right? But because it's nighttime, the cones which are here, they're not contributing to your night vision. The rods are. But if you're looking straight at that star, the light from the star isn't hitting an area where there are lots of rods. So actually, if you look at the faint star and look a little to one side, you'll see it better because now light from that distant star would be hitting an area lateral to the fovea, where now we have more rods and cones. We'll talk in just a few minutes about the bleaching of the rhodopsin here. So like if you were using your night vision and someone shone a flashlight in your face, then that would cause all of the retinol to dissociate from opsin, and you would not be able to see well until it returned. And that then, takes a while. So you would notice like maximally 45 minutes, but, it would take you 10 minutes, 20 minutes. And over this time, your night vision would be becoming better. So we use our rods to see in dim light, and it does not see in color. And it is best when we don't look directly at it because there are more of these rods in the lateral portions of our eye. They also contribute to other things. So for example, someone said, I saw something move out of the corner of my eye. So obviously just, being able to detect movement, in the corner of our eye is also important as well. There is a second type of photoreceptor called cones because of the outer segment having a cone shape. And there are three different kinds of cones. One is maximally sensitive to blue light, one to red, and one to green. Now, there's no magic number of cones, and organisms vary. So a lot of things can see ultraviolet that we can't. So a bumblebee approaches a buttercup, which to you looks like a yellow flower, but there's actually ultraviolet patterns on it that you can't see. Some fish can see ultraviolet light, some birds can, bees can, you can't. And so there's nothing, magic about these, three colors. Most mammals have blue cones and then another cone which perceives red and green. So most mammals are dichromatic, where they can see those two different color modalities. And that's why, say, hunters can wear orange in the woods, because deer don't perceive the difference between orange and green. Now, for primates, color vision is an advantage. Obviously, distinguishing between a red fruit and a green leaf can help a primate. And so the primates then duplicated a gene so that instead of just detecting red and green, there were now two of these genes on the X chromosome, and then one specialized for red and one specialized for green. So we are trichromatic. So some cones express the blue opsin and are responding to blue light. Some are expressing the red opsin, some the green opsin. These are separate genes that we have. All right, and so we have these cone-shaped photoreceptors. They are most densely packed in the fovea, and this is the area where we have the greatest visual acuity. So when you look straight at something, the light that you're looking straight at hits the area which is richest in cones, this fovea, and so that's where we have our best vision. These cones work in bright light. So in the middle of the day, you're very good at distinguishing between colors. But then when it gets to dusk, you start to see this color less, because now the rods are becoming predominant less in terms of cones. All right, so this is what we use for color vision. I mentioned that there were then 3 opsins for different shades of color. Not being able to make one of these would make you colorblind, but thus, since there's 3, there's different forms of color blindness. 2 of the genes for red and green, are on the X chromosome. So they are sex-linked, and therefore color blindness is more common in men than in women. Now, the eye in some ways is built a little backwards. And so let's say here is the direction that light is coming here, and red, green, blue light, etc. It's coming here. Well, it would make sense then to have the photoreceptors, the rods and the cones, here, ready to greet the light when it came, rather than having the light then having to filter through other layers. So there are, that would make sense. And then if the rods and cones were here, facing here with their outer segments, then the axons, et cetera, could then project this way towards the brain. But that's not the way that the eye is built. So when light hits the retina, it doesn't hit the part where the rods and the cones are. All right, and so it has to then filter through a number of layers with other cell types before it hits the rods and the cones. And even then, it hits the wrong end. It doesn't hit the part with the photoreceptors. So instead, it then has to proceed further to reach those. So great, we've hit the wrong part of, so the eye is kind of built backwards in that sense. All right, well, now we've detected light. All right, well, the brain is this way. So you'd think, well, all right, once we detect light, now axons can go towards the brain, right? No. So then it's going to go back the way we came, out towards the lens again. So as you look at where this, the light, once we've detected it, then it gets processed, and we send the information through these other layers, which I'll mention in a second, to these sensory neurons, which will form the optic nerve. So the information is now going away from the brain back towards the lens. But we then collect the information here, all of these axons, form an optic nerve, but then we have to then have it go back. If the brain is up here and now the information is here, we have to have it then go back to the brain, back through. And so for those of you who are following along, so the light went this way, the information about the light that went this way, and now we collect it and we have to send it back that way. And so therefore, where does that happen? Well, it happens here. So here, there is an area where all of these axons then have to go through the retina. Now, you wouldn't have to have this, if the eye was constructed differently, but because it's made the way it is, these axons have to go this way. So there's going to be an area where all of the axons bundle together and form an optic disc where the optic nerve forms. So the optic disc is where the optic nerve is forming all of these axons and bundle together, and they cross through the retina. But the problem then is, if this is where a nerve is forming, you don't have photoreceptors here. In other words, you have a blind spot. All right. So if you look at your anatomy textbook, for example, there'll be a page where you'll see like a dot and an X. And so there are instructions, like if you hold the your book out from you and you look at the X, at a certain point, as you look at the X, you'll see the dot, you'll see the dot, you'll see the dot, and all of a sudden, oh, the dot's gone. And actually, you just see a blank page. Now the page isn't blank, there's a dot there, but you don't see it. You just see a blank page. And then when you bring it closer, the dot reappears. What happened? Well, when you bring the book closer to your face, when the light from the dot hits this area where you don't have photoreceptors, because this is where your optic nerve is forming, this then, without photoreceptors, this is a blind spot. And your brain just kind of fills it in at random. So your brain actually plays with your field of vision. So one thing, since lenses flip objects, the light from my eye, the light from me is actually hitting your brain or your retina upside down. So I look like I'm upside down when it hits your retina, and your brain just flips it, so that you could walk around better. But you don't actually see the light from my forehead on the top part of your retina. You see that on the bottom part of your retina, and your brain just flips it. So your brain kind of messes with your field of vision that way. And then it also messes with your field of vision because, once again, you have a blind spot and your brain just imagines what would be there and just fills it in from whatever the surrounding area is. And so when you bring this closer, because most of the page is blank, once the light from that dot hits here, your brain just says, all right, the rest of the page is blank, I'll bet you this area is as well. And it just fills it in there. So we have a blind spot where the light from our optic disc hits. Okay. So there are multiple cells in the retina. So that we pay a lot of attention to the rods and the cones. The rods, once again, are for perceiving things in black and white and gray. In dim light, the cones are for perceiving color. But then once that information, is projected here, it then the rods and cones synapses with other cells, which then start processing the information. So we have horizontal cells, we have these bipolar cells, so-called, because they have two processes. Now, this gets very complicated very quickly. I'm just going to make a couple quick points and move on. One of which is, notice how the cones tend to synapse with only one single bipolar cell, okay? Whereas this rod is synapsing with this bipolar cell, but this bipolar cell also then synapses with this rod and that rod and that rod. This means that our night vision is more sensitive, because I will say, I see a little light if light hits this rod, this rod, this rod, or this rod. Any of those rods will then stimulate this bipolar cell. So that's now very sensitive. I have a wide area over which I'm trying to detect light. However, it means my night vision isn't going to be very precise. It's going to be blurry. If you're looking for nice, crisp, edges to the things that you're looking at, you want a setup like this one, where each photoreceptor is interacting with one bipolar cell. All right. So there's processing, which goes on here. And then there are other cells as well. So there's these amacrine cells, which are involved as well. And then the ganglion cells, and it's these cells which then have the axons which form the optic nerve. So there are multiple layers of cells in the retina. Once again, this gets very complicated very quickly. I just want to point out one other thing though. Some of these cells are actually, they have opsins as well. They actually detect light. So these are not the only cells which are detecting light, but these are the only cells which do vision. All right, well, if these cells are detecting light with different opsins, what are they doing? We're not seeing with them. Well, that's true, but there's a lot of things that you do with vision other than, seeing. So there's vision, but then you just want reflexes. Like if something's coming close to your eye, like if if there's a shadow or something, you might want to blink. Right, that doesn't have to form a visual image. That's detecting light, etc. And another thing is we want to keep track of the day and night. We want circadian rhythms, so that at night, say you make a lot of melatonin that does things in your body, but in the day you're not. So you want to keep track of how long the day is, etc. So that's actually being done by some of these other cells. They are detecting light, but not for vision. Once again, light is important for a lot of things, not just for vision. And I apologize. I just wanted to run back on that cow and just say something like when biology students study, the microscope, they say, oh, this is the lens, and what lens is like the lens of your microscope. What it would do is it inverts an object. And somewhere, I think it's this video, if not, I just make the point that once again, your lens is a lens, just like that microscope lens was. And just like the microscope inverts things so that when you move the slide right, it looks like it's moving left. And when you move the slide away from you, it looks like the image is moving towards you. The light from this cow, if you're asking how the image of the cow hits the retina, it's actually upside down and backwards. So this is a lens, just like the lens of the microscope that inverts things, and our brain just switches it, as I said. Okay. Let me just say something about the actual molecules which are detecting light before we go to the parts of the brain which process it. So rhodopsin, let's talk about the opsin in rods. We have that name because there's a protein called an opsin, right? It's a G protein coupled receptor. And then it binds to something known as retinol, which is derived from vitamin A. That's a reason vitamin A is important. If you don't have enough vitamin A in your diet, as unfortunately many people in the world do not, you can develop night blindness, which can actually then become full-fledged blindness at some point. So the opsin then passes through the membrane where it can interact with a G protein called transducin. Right, the reason this is called a G protein coupled receptor is because it binds with transducin. Okay, and so I'm sorry, I think I skipped a so the key is that retinol has two forms. All right, one form is bent, and when it's bent, it can stick on opsin and form rhodopsin, which is now a complex that has both the retinol and the opsin. But then when it straightens, then it dissociates from rhodopsin. So this retinol has two forms, a cis form, all right, because of this double bond between carbons 11 and 12, or this could then change into the trans conformation here, that's straighter. The cis form can stick to opsin, all right, the trans form doesn't, and it'll stick to something else, as we'll see. And so when light hits retinol, this molecule, this portion of it rotates from the cis form to the trans form. And this then causes the molecule of retinol to dissociate from opsin. This is what's called bleaching, all right, because now this pigmented molecule is leaving. It's bleaching, all right, and then it instead of being called retinol, we call it meta-rhodopsin or meta-2, and I'll talk about what it sticks to in just a second. So after it sticks to light, this then dissociates. Now, we'll package it back together, but in mammals, that takes about 45 minutes to make more cis retinol that can stick again. So that's why if someone shines a flashlight in your eyes, that causes all of this bleaching, all of the retinol dissociates. And now it takes you a while to make more attached to opsin so that you'd be able to detect light again. When this does form that transform, the meta-rhodopsin, it then binds to the G protein, transducin. Okay, so the G protein normally has these two parts which are attached to rhodopsin, but when meta-rhodopsin binds to it, the G protein separates, all right? So the key then is, I detected light, all right? Retinol bound light. That was an external stimulus, external to the cell, but then the rhodopsin dissociated, formed meta-rhodopsin, which caused the G protein to dissociate. And that's now going to cause changes inside the cell. It's a second messenger system. We don't have to get into that, but this video goes into it. It activates something called photodiesterase, which then changes cyclic AMP, or my fault, cyclic GMP. So you notice that there's a ring here because of how the phosphate attaches. So it's a cyclic form, cyclic GMP. This will then change to make a form of GMP which isn't cyclic. All right, so light hit caused meta-rhodopsin to stick to a G protein, which then split up, which activated phosphodiesterase, which then caused cyclic GMP to become GMP, which then changed ion gates. Remember that some ion gates will be either open or closed based on what molecules can bind to them, and cyclic GMP is one of them. So when cyclic GMP changes, then that changes these ion channels. Cyclic GMP will open the channels and let sodium come in, but now that cyclic GMP has changed, the ion channels close. Sodium channels close. This changes the depolarization of the cell. All right, so now if sodium isn't coming in, the cell hyperpolarizes. That then changes calcium levels. Some calcium are voltage, some calcium gates are voltage regulated. So the amount of sodium coming in changes the calcium gates. And then calcium is needed for the exocytosis of neurotransmitter. If you're a typical student, I've already lost you. But the point is that light causes a change in retinol, which causes a number of changes in the G protein, which causes the activity of phosphodiesterase, which changes the amount of cyclic GMP, which changes whether the sodium gates are open, which changes whether the calcium gates are open, which changes the amount of calcium, which changes the release of neurotransmitter. But the neurotransmitter is now telling the brain, I've detected light. So all of this, what it's done is allow us to link the signal of light to a change in neurotransmitter levels, which then lets the brain know that we have detected light. Just one last thing, then, if you were to look at rhodopsin, I just want to look at rhodopsin, we could look at the cones, but once again, I think this is enough molecular for the average student. So if you were, let's look at a rod in the dark versus in the light. In the dark, we have cis retinol, which is bound to opsin to form rhodopsin. And as a result, then we have cyclic GMP, which is opening sodium gates. Sodium is coming in, and the cell is depolarizing. But then when light binds this causes the cis retinol to become trans-retinol, which then as metal-rhodopsin binds to the G protein, now we have GMP rather than cyclic GMP. The sodium gates close, the cell hyperpolarizes, we open calcium gates, and that then changes neurotransmitter production. Okay, so now the ganglion cells, which are forming the optic nerve, are carrying an electrical message about the light that we have detected. So from the retina, these axons then all bunch together in that optic disc to form the blind spot. And then from there, this optic nerve then goes behind the eye through the sphenoid bone. The sphenoid bone has a hole, the optic foramen, or the two of them, the optic foramina. This is right in the sella turcica region of the sphenoid bone. The optic nerve then passes through that hole, and then it crisscrosses here at what's called the optic chiasma, the area where the optic nerves crisscross. And thus most, but not all, of the information from your right eye will go to the left half of your brain and vice versa. After the optic chiasma, this is known as then the optic tract, which will then go to places of your brain, which I'll mention in just a second. This is the second cranial nerve. The first cranial nerve is the olfactory nerve, which goes through those openings. The optic nerve is the second cranial nerve. And obviously damage to this nerve can cause a number of visual deficits, including blindness. So the optic tract then enters the brain, and the light can, be processed in a number of regions. In the midbrain, which develops from the mesencephalon of the embryonic brain, the midbrain has these bumps. Now, the inferior bumps are what we call the inferior colliculi, and that processes hearing. But then above that are the superior colliculi. These bumps, that's a bump, there's a bump. The superior colliculi, these then process vision. Now, a lot of people don't care about the midbrain or the thalamus because, they care about the parts of the brain that we are consciously aware of. But there's lots of things, lots of involuntary processing which occurs in vision. Vision is quite complex. Plus, there's a lot of reflexes. When something comes to the eye, we blink, or as we track objects with the eye, or move the head to face the eye. So the midbrain has these bumps processing hearing and vision, and these cells, which can then send motor commands like movements of the head. So we can, we hear a sound and we try to look to see what that is, or we move our head along with something, that we're tracking a visual image of. So the superior colliculi of the midbrain receive input from those optic tracts, and then projects to the thalamus of the brain. So the medial geniculate nucleus is getting input from the inferior colliculus, where it can process hearing. But meanwhile, there is a lateral geniculate nucleus of the thalamus. This is getting input from the retina directly, but then also the superior colliculus. So then here the thalamus is also participating in involuntary processing, and the thalamus here, so the superior colliculus would be here of the midbrain. The thalamus has a lateral geniculate nucleus that then receives input from here, and then this then projects to the visual cortex. The visual cortex is the part of the cerebrum where we are consciously aware of vision. It's in the occipital lobe. So here's the visual cortex here. So the occipital lobe is here, all in the green. The visual cortex is the area where we're just kind of getting information about vision. So you're seeing things. But then around the visual cortex, we have what's called the visual association area, because we don't just see things, we interpret. We realize, oh, that's, a dog, that's a tree, that's the face of a friend of mine, while that's a face of someone that I don't know. A stroke that affects the visual cortex would mean that we can't see. It's like, oh, I see nothing, it's just darkness. Whereas a stroke which might affect the visual association area, that would result in a deficit in interpreting vision. We would still see, we just wouldn't recognize the face of someone that we used to know, etc. Also note that this vision is a special sense. Unlike the general senses like touch, where we feel it all over the place, and they all go to this somatosensory cortex, vision is only perceived in one place, the eyes, and it goes to its own special region of the cerebrum. So the occipital lobe is just for vision, unlike touch and pressure, et cetera, which goes here. And then one final thing I mentioned, when we think of the eye, we're thinking of vision, but there is more to detecting light than just vision. One of the very important things that we do is we know when it is light or dark. And we have these circadian rhythms. Circadian means about a day, circadian. So there is a sleep-awake cycle, but there's also daily changes in temperature, hormone secretion, hunger, autonomic function. This also depends on light. So from our optic chiasma, we do have axons which project to a different part of the brain, which is the suprachiasmatic nucleus of the hypothalamus. And this is the master clock of the body's circadian clock. The suprachiasmatic nucleus then projects, so here's a light from the eyes, then goes to the suprachiasmatic nucleus of the hypothalamus. Here's the thalamus. This area underneath it would be the hypothalamus. This then goes to a ganglion, and then that returns then to the pineal gland from that superior cervical ganglion. And the pineal gland makes melatonin at night. All right, and so all of the cells of your body know whether it's light or dark out, because there's melatonin in your blood when it's dark. There's not melatonin in your blood when it's daytime. And so what you've managed to do in the hypothalamus is to convert an external stimulus of the presence or absence of light to an internal stimulus, the presence or absence of melatonin. And so once again, the eyes do a bit more than just do vision, and this was one example of that.